welcome to this Anatomy Physiology Bio 202 video lecture on the urinary system. I'm Mr. Kennedy, and I'll be your guide as we explore this topic together. The urinary system consists of the kidneys, ureters, bladder, and urethra. Its purpose is to maintain homeostasis by managing the volume and composition of our fluid reservoirs, primarily our blood. This is a diagram to illustrate the basic anatomy of the urinary system in a female. Please note the relative positions and names of each labeled item in this diagram. Homeostatic kidney functions. Homeostatic kidney functions include the regulation of blood ionic composition with regard to sodium, potassium, and chlorine ions, the regulation of blood pH, the regulation of blood volume, and the regulation of blood pressure. The homeostatic kidney functions also maintain blood osmolarity and produce the hormones calcitrol and erythropoietin. Kidney functions also include the regulation of blood glucose levels, and finally, the excretion of metabolic wastes and foreign substances, such as drugs and other toxins. This is a graphic that illustrates the kidney's proper anatomical location. Note the position of the kidneys and each labeled item in this diagram. The kidneys are retroperitoneal and partly protected by the lower ribs. Renal anatomy. The indented area in the kidney is called the hilum. This is the entrance for the renal artery, renal vein, ureter, nerves, and any lymphatics. The external layers of the kidney are made up primarily of connective tissue. Moving from superficial to deep, we have the renal fascia, adipose capsule, and renal capsule. The fascia anchors to other structures, the capsule protects and anchors, and the renal capsule is continuous with the ureter. The internal renal anatomy starts with the renal cortex, which is the outer layer, then the renal medulla, which is the inner layer. Next are the renal pyramids, which are the secreting apparatus and tubules, and the renal columns, which anchor the cortex. Here we have a cross-sectional diagram of the kidney. Note each labeled structure and review the other pieces of the kidney mentioned on the previous slide using this diagram. Here we have a slightly more detailed cross-sectional picture of the kidney to illustrate the papillary ducts. The papillary ducts empty urine into the calyces. The calyces pass urine to the ureter. Blood and nerve supply of the kidney. Blood supply. Although the kidneys constitute less than 0.5% of our body mass, they receive 20 to 25% of resting cardiac output. Your nerve supply to the kidneys occurs via renal nerves. Renal nerves primarily carry sympathetic outflow. They regulate blood flow through your kidneys. The nephron. The nephron is considered the workhorse of the kidney, or, put another way, the primary filtration unit of the kidney. Note each labeled structure in their position on this diagram on this slide. The renal corpuscle filters the blood plasma, while the renal tubule modifies the filtrate to urine. The renal corpuscle consists of two parts. The glomerulus is the first part. The glomerulus is a mass of capillaries. The glomerular, or Bowman's capsule, which has a visceral layer of podocytes which wrap around the capillaries, is the second part. The glomerulus, as a mass of capillaries, is fed blood by the afferent arteriole and drains into the efferent arteriole. Mesenchymal cells are contractile and help regulate glomerular filtration. The Bowman's capsule or glomerular capsule has a visceral layer of podocytes which wrap around the capillaries. The filtrate is collected between the visceral and parietal layers. This is an illustration of the renal corpuscle. Again, note the position of each labeled structure in the diagram. Next, we have the glomerular endothelial cells. The glomerular endothelial cells have large pores or fenestrations and are considered leaky. Basal lamina lies between the endothelium and the podocytes. The podocytes form pedicels, between which are filtration slits. This is an illustration of those aforementioned structures. The filtrate passes from the glomerular capsule to the renal tubule, as illustrated here. From there, we go to the proximal convoluted tubule, the nephron loop descending, followed by ascending, and the distal convoluted tubule. The ascending loop of the Juxtaglomerular Apparatus contacts the afferent arteriole at the macula densa. The wall of the arteriole contains smooth muscle cells, or juxtaglomerular cells. The apparatus regulates blood pressure in the kidney in conjunction with the autonomic nervous system. Here we see that structure illustrated in this diagram. The distal collecting tubule and collecting duct are illustrated here to the right. The principal cells in these structures have receptors for ADH and aldosterone. Intercalated cells in these structures help to manage blood pH. There are two kinds of nephrons. The cortical nephrons, which make up 80 to 85% of nephrons, which contain a renal corpuscle in the outer portion of the cortex, short loops of Henle extended only to the outer region of the medulla, and who create urine with osmolarity similar to blood. And second, the juxtamedullary nephron. Juxtamedullary nephron has a renal corpuscle deep in the cortex with long nephron loops. They receive blood from peritubule capillaries and the vasa recta. Their ascending limb has thick and thin regions, and they enable the kidney to secrete very concentrated urine. Here we see each illustrated on this slide for your comparison. Renal physiology and urine formation involves glomerular filtration, tubular reabsorption, and tubular secretion. Each process is illustrated in a simplistic diagram seen here. Glomerular filtration is driven by blood pressure. It's opposed by capsular hydrostatic pressure and blood colloid osmotic pressure. Water and small molecules move out of the glomerulus. In one day, 150 to 180 liters of water pass out into the glomerular capsule. The glomerular filtration rate is the amount of filtrate formed by both kidneys each minute. Homeostasis requires kidneys to maintain a relative constant GFR. Too high, and substances will pass too quickly and are not reabsorbed. Too low, and nearly everything will be reabsorbed, and some waste products will not adequately be excreted. This diagram illustrates the process of glomerular filtration and the relative pressures in each area. GFR averages 125 mils per minute in males and 105 mils per minute in females. It's controlled by renal autoregulation, neural regulation, and hormonal regulation. Renal autoregulation begins with myogenic mechanisms. Myogenic mechanisms involve smooth muscle cells in afferent arterioles who may contract in response to elevated blood pressure. Tuboglomerular feedback is also part of the renal autoregulation process. In tuboglomerular feedback, high GFR diminishes reabsorption, macula densa inhibits release of nitric oxide, and your afferent arterioles constrict. Tuboglomerular feedback is a negative feedback loop as seen illustrated on this slide. Neuroregulation involves the kidney's rich supply of sympathetic nerve fibers. Strong stimulation, exercise, or hemorrhage will cause afferent arterioles to constrict. Urine output is reduced and more blood is available for other organs. Hormonal regulation involves the use of hormones such as angiotensin. Angiotensin II constricts afferents and efferents, diminishing GFR. Atrial natriuretic peptide, or ANP, relaxes mesangial cells, increasing capillary surface area and GFR. ANP is secreted in response to stretch of the cardiac atria. Much of the filtrate is reabsorbed, especially water, glucose, amino acids, and ions. Secretion helps to manage pH and rid the body of toxic and foreign substances. Here we can see the plasma, filtrate, and urine compositions. Note each and its relative volume and rate of production. Much of the filtrate is reabsorbed by both active and passive processes. Again, especially water, glucose, amino acids, and ions. Secretion will help to manage pH and rid the body of toxic and foreign substances. Reabsorption routes. The paracellular reabsorption route is a route that involves the passive fluid leakage between cells. The transcellular reabsorption route is a route that moves directly through the tubule cells. Here we can see our reabsorption routes illustrated on this slide. Note which processes occur passively and which processes occur actively. Primary active transport. Primary active transport uses ATP like sodium-potassium pumps. At rest, primary active transport accounts for 6% of the total body ATP use. Secondary active transport is driven by ions' electrochemical gradients. Symporters move substances in the same direction, while antiporters move substances in opposite directions. Water reabsorption. Obligatory water reabsorption accounts for 90% of the water that we absorb. Water follows the solutes that are reabsorbed. Facultative water reabsorption accounts for the other 10%, and it's regulated by ADH, or antidiuretic hormone. Reabsorption and secretion in the proximal convoluted tubule. Sodium, and using glucose symporters, sodium and hydrogen ion antiporters are used in the proximal convoluted tubule in the process of reabsorption and secretion. Aquaporins are membrane proteins that are permeable to water. Here we see the transport mechanisms of symporters and antiporters in the proximal convoluted tubule. Here we see the passive reabsorption in the late proximal convoluted tubule. Reabsorption in the loop of Henle. The loop of Henle is relatively impermeable to water, especially the ascending limb. Little obligatory water reabsorption occurs here. More commonly are the sodium, potassium, and chlorine symporters. This graphic illustrates the reabsorption in the nephron loop. Sodium and chlorine symporters reabsorb ions in the distal convoluted tubule. PTH stimulates the reabsorption of calcium in the distal convoluted tubule. It also inhibits phosphate reabsorption, enhancing its secretion. Late distal convoluted tubule and collecting duct are made up of principal cells and intercalated cells. Principal cells have sodium-potassium pumps to reabsorb sodium ion and aquaporin, which again are stimulated by antidiuretic hormone. Intercalated cells reabsorb potassium and bicarbonate and secrete hydrogen ion. Regulation of water reabsorption by ADH is governed by a negative feedback loop, which you can see illustrated here. Urine production. Fluid intake is highly variable. Homeostasis requires the maintenance of fluid volumes within specific limits, and urine concentration varies with antidiuretic hormone. High intake will lead to dilute urine and high volume. Low intake will lead to concentrated urine of low volume. Formation of dilute urine. Your glomerular filtrate and blood have the same osmolarity of approximately 300 milliosmoles per liter. Tubular osmolarity changes due to the concentration gradient in the medulla. When dilute urine is formed, the osmolarity in the tubule increases in the descending limb, decreases in the ascending limb, and decreases more in the collecting duct. This is an illustration of the changes in tubule osmolarity for the formation of dilute urine. The thick ascending limb has symporters which actively reabsorb sodium, potassium, and chlorine, and low water permeability. Solutes leave and waters stay in the tubule. The collecting duct has low water permeability in the absence of antidiuretic hormone. Again, here we see the changes in tubule osmolarity for the formation of dilute urine. Juxtamedullary nephrons with long nephron loops are needed for the formation of concentrated urine. Here, the osmotic gradient is created by a countercurrent multiplier. Solutes are pumped out of the ascending limb, but water stays in the tubule. The medulla osmolarity is increased. This is an illustration of the formation of concentrated urine. In the presence of antidiuretic hormone, collecting ducts become very permeable to water. Tubular fluid there becomes very concentrated. The movement of water also carries urea into the medulla, contributing to its osmolarity. Again, we see that illustrated in this diagram. The countercurrent exchange. Here, the loop and duct cells require nutrients and oxygen from your blood supply. Capillaries that feed them, also known as the vasa recta, form loops like those of nephron loops in the medulla. Incoming and outgoing blood will have the same osmolarity, and this maintains the medulla concentration gradient. Here we see that illustrated on the side of the diagram with the capillary. The evaluation of kidney function is done using routine urinalysis. Routine urinalysis primarily evaluates for the presence of abnormalities in the urine, such as albumin, glucose, red blood cells, ketone bodies, and microbes. Urine transportation and storage. Each ureter transports urine from a renal pelvis by peristaltic waves, hydrostatic pressure, and gravity. No anatomical valve is present at the opening of the ureter to the bladder. When the bladder fills, it compresses the opening and prevents backflow. The bladder is hollow, distensible, muscular organ with a capacity averaging 700 to 800 mils. Here we see the basic diagram of the urinary bladder. And finally, we have a comparison of the male and female urethras, the tubes that lead urine out of the body. And this concludes our coverage of the urinary system.